Genome editing is an exciting new technology due to its ability to change how an organism's DNA is expressed.
By adding, removing, or altering the genetic information or genes at particular locations of an organism's genome, scientists are able to control what genes to express or suppress. The most promising technology in genome editing is CRISPR-Cas9. CRISPR-Cas9 has revolutionized the genome editing field because it is faster and more efficient, as well as more accurate than other gene editing methods. Although the technology is almost 10 years old, the first treatment using CRISPR has just been approved by the FDA for human testing. Sickle cell anemia is a genetic disorder that is caused when an individual inherits a hemoglobin S gene from both parents, rather than inheriting the gene from only one parent, as individuals without the disorder do. Those who carry this form of anemia may have deformed red blood cells that resemble a sickle. These deformed cells can potentially get stuck inside the blood vessels and ultimately restrict the flow of oxygen and nutrients to key organs. Using CRISPR technology, scientists are able to block the gene responsible for causing the sickle-shaped mutation in red cells. They do this by extracting the patient's natural stem cells, which is the bone marrow cells that generates all the body's red blood cells. These cells can be harvested for gene editing outside the body. After these cells are removed, the remaining bone marrow is destroyed with chemotherapy to allow space for the edited stem cells to grow and eventually completely replace the red cell with the unwanted mutation. After 6 years of work, that experimental treatment has now been approved for clinical trials by the U.S. Food and Drug Administration. This is without a doubt the beginning of gene editing therapy.